Hi, everyone. My name is Wei, founder and lead analyst at Homodeus Research. Thank you for taking the time to join us today. Today, I'm delighted to be joined by Dr. David Stamler. Hi, Dave.
Good morning, Mike.
Good morning. So, Dr. David is the managing director and CEO of Alterity Therapeutics. The ticker is ATH. They've got a market cap of around $110 million. So Dr. Stamler has spent decades developing therapies for neurodegenerative diseases and has led Alterity's clinical development of ATH434, a potential disease-modifying treatment for multiple system atrophy, or MSA, a rare and devastating neurological disease for which there are currently no approved therapies. So before we go in, I'd like to highlight 3 reasons why now is an exciting time to be looking at Alterity. Firstly, Alterity now has established a clear regulatory path to phase 3 after reaching agreement with the FDA on the design of its pivotal registrational study. This is one of the most significant de-risking events for any clinical stage biotech. Secondly, the company recently strengthened the commercial value of ATH434 by securing a new US composition of matter patent, protecting the drug through at least 2045. And thirdly, Alterity is entering a catalyst-rich period as it prepares to initiate its pivotal phase 3 trial, with multiple regulatory, operational, and clinical milestones expected over the coming year. So with that, I'll hand it over to you, Dr. David Stamler. David, thank you very much for joining us today.
Well, thank you, Wei, and thanks everyone for joining the call today. Wei, as my presentation gets loaded, perhaps you can confirm it's in full screen mode and not in slide sorter mode?
Yes, we can see it in full screen mode now. Thank you.
Wonderful. Well, thank you. And what I really hope to tell you about today is a disease you've probably never heard of, but by the end of this talk, I expect you won't forget it. It's quite a devastating condition, and there are no approved therapies for it. These are our forward-looking statements. I encourage you to read these on our website. So alterity is actually a word in the English language that means the state of being different. And this really embodies our mission. What we're trying to do is to change the state of individuals living with neurodegenerative diseases from disease to health. And we're really trying to do that by slowing the course of disease progression. And that's really what I hope to share with you over the next several minutes. As mentioned, we are developing disease-modifying therapies, and we are targeting a first-in-class therapy for a disease called Multiple System Atrophy, or MSA. Earlier in 25, we disclosed compelling efficacy results in our phase 2 study, demonstrating slowing of upwards of 50% disease progression when compared to placebo on an endpoint that the FDA and other regulatory authorities recognize as the endpoint to support an approval. This was done in the context of a favorable safety profile, so really a very important profile from an efficacy and safety standpoint. Next, as mentioned, MSA is a rare disease, therefore it qualifies for orphan drug pricing in the US and in other regions. And because there is nothing approved for this devastating condition, we conservatively estimate that upon successful approval of the drug, we would expect peak sales of upwards of $2.5 billion US dollars. And that's actually going to increase substantially with the patent that was previously mentioned. Our drug is a unique molecule that really targets excess iron that drives the pathology of this disease and others, including Parkinson's disease. I will be sharing some of those data, the background, to help explain how we target this excess iron. And finally, as Wei mentioned at the beginning, we recently completed a successful end of phase 2 meeting with the FDA, where we reached alignment on the critical aspects of our phase 3 program so that we now have a blueprint for the path forward. This is an outline of what I'll be talking about today. Most of the time, I'll be speaking about our lead indication, multiple system atrophy, which are these three studies that you see here in blue. But not far behind is an emerging interest we have in Parkinson's disease. Parkinson's disease, as many people know, is the second most important neurodegenerative disease following Alzheimer's disease, and there are no therapies approved to slow the course of that disease. So this new patent that we got will enable us and our prospective partners to pursue a Parkinson's indication down the road. And we also have potential to pursue other orphan indications as listed on the slide. So the drug has many options for treating neurodegenerative diseases. Now, turning to multiple system atrophy, or MSA for short, we refer to this as a Parkinsonian disorder. Now, that's one side of the coin, and I think everybody knows what a Parkinson's disease individual looks like, someone who's got the disease. However, the other side of the coin in MSA is that it's far more aggressive along the lines of similar to ALS. So even though it can look like Parkinson's disease early on, it progresses very rapidly, as shown in this figure on the right side of the slide. So patients can present with nonspecific symptoms that you see listed here. And then after a couple of years, they often progress to develop motor symptoms of Parkinsonism. That's the stooped posture or the shuffling gait or the tremor, as well as unsteady gait and frequent falls. And then after those neurologic symptoms present, patients often progress to require a cane or a walker. And then more than 50% of individuals with MSA require a wheelchair within 5 years of symptom onset. And the disease has a median survival of just 7 to 8 years after symptoms first appear. So it really is a devastating condition, and there is an urgent need to find a novel therapy for this. So I'm going to tell you a little bit about how our drug works. But before I do that, I do want to share maybe the outlines of the key pathology that's involved in both MSA and Parkinson's disease. On the top of the slide, you see what's this colored protein that we refer to as alpha-synuclein. And alpha-synuclein is present in all neurons within the human body, and it's necessary for neurons to communicate. However, in diseases like MSA and Parkinson's disease, this protein forms aggregates. And when the protein aggregates, the body tries to clear it, which often elicits an immune response and inflammation. And it also, because it aggregates, the protein can't function, which leads to impaired neuronal communication that underlies the symptoms that the patients present with. The other thing that we target with our therapy is iron. Now, you probably remember from your high school biology that iron is important for energy production or for oxygen transport. But within the central nervous system, iron is critical for key cellular functions, notably neurotransmitter synthesis, dopamine, which you've probably heard of. And iron is also critical for synthesizing myelin, and that's the fatty sheet that wraps around the nerve terminals that allows them to have rapid neural transmission. For a variety of reasons, there is excess iron that accumulates in the central nervous system of patients with these diseases. And it really drives this alpha-synuclein protein to aggregate. And this is really what we're trying to address with our therapy, both the misfolding protein and the excess iron. This is an autopsy study that looked at the brains of individuals who died from either Parkinson's disease on the left or MSA on the right. And what we see in both cases is that the patients who are in blue have excess iron in regions of the brain where there is pathology. So we see in Parkinson's disease, the substantia nigra, these two regions here, there's elevated iron compared to controls. And the same thing is true in MSA, although many other brain regions are affected. And this is really the iron that we're trying to address with our therapy. What we're trying to do is reduce the pathology that this excess iron causes. Now, this slide really kind of outlines how we approach this. As mentioned, there are various reasons that there is excess iron within the central nervous system. Sometimes inflammatory cells cause the release of iron. And when you have this excess iron present in the central nervous system, it actually causes the alpha-synuclein to aggregate, if you see in this dark blue arrow on the bottom. But it also generates free radicals, and those free radicals actually damage intracellular structure like mitochondria and DNA. And they really form the basis for the injury. And then that oxidative stress actually causes the alpha-synuclein protein to aggregate. So you really get this vicious cycle. And we're really trying to break this cycle with our therapy by redistributing the excess iron within the central nervous system. So to tie this all together, our drug, which I'll tell you about in a moment, really acts as an iron chaperone to redistribute the excess iron in the central nervous system. It then leads to reduced aggregation of that important protein synuclein, as well as reduced oxidative injury. It leads to preserved neurons and their support cells called oligodendrocytes, with the overall goal of stabilizing function or slowing its decline. Based on this mechanism, we believe 434 is a potential disease-modifying therapy and something that is not been, no such treatment has been approved for MSA or Parkinson's disease. All right, so now here's our molecule, ATH434. It's referred to as a small molecule, which means it can be orally administered and absorbed through the GI tract. Now, this is obviously a preferred route by patients and doctors. Some therapies that are in development in this area are actually intravenous injections, or they're injections directly into the central nervous system, things that are difficult for patients to tolerate. Our drug has been shown to readily cross the blood-brain barrier, which is important so it can get to the site of action. And it also has been shown to cross across the cell membrane, so it can address the pathology that's ongoing inside the cells. As mentioned, the drug acts as an iron chaperone, which means it can bind to iron with moderate affinity and then shuttle it around the cell so that it can be stored or buffered or even removed from the cell in some cases. And this is really how it does it. And it does it nicely in a way that does not lead to iron reduction or anemia associated with other drugs that are designed to remove iron from the body. The drug, as mentioned, has broad treatment potential. And regarding regulatory status, we have not only orphan drug designation in the US and in Europe, but also fast track designation in the US. Okay, so now let's turn to the clinical development program for ATH434 in MSA. Now, I'll be spending my time talking about these few studies, but most of our time I'll be talking about phase 2. Before we talk about that, a unique aspect of our program is this natural history study called BioMuse. And we did this study back starting in 2020. And we did this with the aim of helping us design and de-risk the phase 2 study. We evaluated 21 individuals that had clinical criteria for MSA. And the goal was to identify biomarkers to improve the accuracy of patient selection for these phase 2 studies. That was a very, very successful study, and we think that's a large reason why our phase 2 program was successful. Now, what I want to tell you about is the phase 2 trial, also referred to as 201, and that is a randomized, double-blind, a placebo-controlled trial. That's the gold standard of trial designs for demonstrating and evaluating efficacy. So in our study, we enrolled a total of 77 patients who met clinical criteria for MSA. And if you see highlighted in blue, these individuals had elevated brain iron on MRI. Given how our drug acts, that's quite important. And we also demonstrated that they needed to have an elevated level of a protein in their blood called NFL, and that helps differentiate between MSA and Parkinson's disease, which Parkinson's patients don't have elevated NFL. They were then randomized in equal numbers to 75 milligrams twice a day, 50 milligrams twice a day, or matching placebo, and they were treated for 12 months. And the key clinical endpoints are listed here on the right. The most important endpoint is the MSA rating scale, And I'll tell you more about that on the subsequent slide. However, we did also look at several secondary endpoints that are outlined here, as well as a biomarker endpoint of brain iron on MRI. This is the MSA rating scale. It's also referred to as the Unified MSA Rating Scale, or UMSARS for short. And the part of the scale that we focus on is called Part 1. And it looks at these 12 domains that are affected in MSA. And you can tell by those that are listed here: speech, swallowing, fine motor skills, walking, falling, even low blood pressure symptoms called orthostatic hypotension. These can have a profound impact on patients' daily activities and their daily functioning. And that's why the FDA does recognize this as the endpoint to support an approval. And indeed, this is our primary endpoint in the phase 3, in our proposed phase 3 study. So this is the key clinical endpoint, the MSA rating scale. It's a rather busy slide, but on the y-axis of this graph, you see the MSA score. On the scale, higher scores are worse. And what we see is over the 52-week treatment period that the placebo patients worsened by about 8 points on the scale. Whereas the two active dose groups, the 50 and the 75 milligram dose groups in blue and teal, declined by anywhere from 35 to 45% less than placebo, which equates to a treatment difference of 2.7 to 3.7 points. Importantly, both those differences exceed the minimal clinically important difference of 1.5 points that's been shown in an independent study. Now, you may be wondering why the treatment effect at 75 milligrams was somewhat lower than 50 milligrams. I didn't have time to go into baseline differences, but there were a couple of differences in this patient population indicating that they had greater severity of disease at baseline, which really predisposed them to a more rapid decline. And then we also looked at these data after we got the blood levels from the study, and we did show that there was a saturation of effect at the 50 milligram dose group. So we do plan on taking the 50 milligram dose group into phase 3. Now, another important endpoint that we looked at in the trial is the swallowing disturbance questionnaire. And again, here what we see are the change in this score over the course of 52 weeks. On this scale, higher scores are worse. And again, we see the placebo patients in gray worsened by about 8 to 9 points, whereas the 50 milligram patients declined by just 1.5 points, and 75 milligram patients declined by about 5 points, with the difference at 50 milligrams being statistically significant. The FDA likes this scale, as do other regulatory authorities, because this is a patient-reported outcome where you hand the patient the questionnaire. And obviously, it looks at 15 important items, such as choking or coughing when drinking liquids, that really can have an impact on quality of life, as well as lead to pneumonia if they have aspiration. So this is an important endpoint. Another important secondary endpoint that we looked at is the orthostatic hypotension assessment, or OHSA. And again, this is a patient-reported outcome, and it is also a questionnaire where patients answer 6 questions to assess their symptoms of low blood pressure when they move from a sitting to a standing position. This is a cardinal symptom of MSA and very problematic symptom. And what we see is that the placebo patients on this scale, again, where higher scores are worse, worsened by about 6 points on average over the course of 52 weeks, whereas those receiving active treatment actually stabilize over that treatment period. So this is, along with the swallowing, is an important secondary endpoint that we plan to include in phase 3. Next, I talked about the immobility that is a cardinal manifestation of patients with MSA as they lose the ability to walk without assistance or even become immobilized. And we looked at their activity in an outpatient setting with what's referred to as a wearable movement sensor that you see pictured on the right lower hand side of the slide. Patients would wear this pendant around their neck for 2 weeks at baseline, and then every 3 months thereafter, they would do the same thing. And then we would compute from that period, a daily step count, or the total number of minutes per day that they would be walking. And then we would look at the change from baseline to the end of treatment. And on this scale, on these measures, obviously activity, lower scores are worse. So if you take step count, we see that on average, these patients lost in placebo about 24 to 2,500 steps per day, whereas those receiving active treatment lost about half that level of activity. So this is quite important if you consider how mobility is such an important activity to maintain quality of life. Similar findings were observed on total walking time, walking periods, or sit to stand transitions. So now let's turn to safety. I think the efficacy of 434 is nicely demonstrated with the data I just shared, but safety is also important. And I think what's noteworthy is that we saw similar rates of adverse events in the 434 treated patients when you compare to placebo. And this is true for all adverse events, as well as for the most common adverse events that are listed on the slide. Importantly, no severe or serious adverse events were attributed to study drug. And given the fact that 434 can bind to iron, we did carefully look at hematologic parameters like blood counts or iron levels, and we didn't see any evidence for toxicity on those parameters. So really, I think a very benign safety profile, which is quite important considering how fragile these patients are. So as we've discussed a couple of times, in June, we did have a successful end of phase 2 meeting with the FDA where we reached alignment with them on the key elements of the phase 3 study design, including the primary endpoint, the population, the treatment regimen, which is 50 milligrams twice a day. We also reached agreement with them on the selection of secondary endpoints, largely the ones that I shared with you, the swallowing disturbance questionnaire, the blood pressure scale that the patients fill out, as well as the clinical global impression of severity. And we also reached agreement with the FDA on statistical methods and the overall size of our safety database. So now to put that in context, this is what the phase 3 study design looks like. We're targeting around 200 patients to be enrolled that have a clinical diagnosis of MSA, no evidence of severe impairment. And in contrast to the phase 2 design, we're actually going to use brain atrophy to select patients. That's really the cardinal finding in the brains of these patients, that they have atrophy in MSA-affected areas. And this yields very similar results in phase 2 when we looked at atrophy versus brain iron, but it's easier to operationalize. Patients are then will be randomized in equal numbers to the 50 milligram twice daily dose or matching placebo and treated for 12 months. And the primary endpoint will be the UMSAR score, the 11-part UMSAR score that I showed you that we used from phase 2. The key secondary endpoints we've discussed, the swallowing scale, the orthostatic hypotension scale, and the global assessment. And then these are just on the bottom of the slide. There are efficacy analyses. Will include various parameters that will help us manage the study population appropriately from a natural history standpoint. The one thing I'll mention, because I'm now going to go on and talk about commercial, is in the interest of time, I haven't gone through the biomarker data, both the iron data and the brain volume data in this presentation. But if for those who are interested, I've given a presentation at the Canaccord conference, and that's on our website, as well as we did a webinar back in April of this year where with our coordinating investigator and an expert in the field. So if you're interested in seeing those biomarker data, you can check there. So now I want to talk a little bit about the commercial assessment. Based on the phase 2 data that I just shared with you, we created a target product profile, and we showed that to 100 US neurologists. And this was an independent evaluation done by a firm who conducted these interviews. Of those 100 neurologists, half were generalists, half general neurologists, and half were specialists. And And overall, there was a very strong intent to prescribe, with more than 70% of neurologists being extremely likely or very likely to prescribe 434. They clearly recognized the substantial unmet need for treating MSA, and they appreciated the mechanism of action of targeting this protein alpha-synuclein in a novel way. Using relatively conservative assumptions on the prevalence as well as the price of the drug, we estimate global sales of $2.4 billion US dollars at peak. So, to sum up, we have really completed important regulatory interactions with the FDA, aligning not only on the phase 3 study design, but also on certain non-clinical and clinical pharmacology aspects. We have also reached agreement with the FDA on key aspects of our chemistry and manufacturing program. So we have those important milestones behind us. We are actively preparing for phase 3, working to identify sites and qualifying those sites for participation. We have successfully manufactured a drug for use in phase 3 and are in the process of tableting that and packaging that for trial activities. And we do expect to initiate clinical trial activities by year end. As mentioned at the outset, we have recently been granted a new composition of matter patent for 434 that really protects the molecule out to 2045, and that has important implications for both MSA and Parkinson's disease. And accordingly, we are evaluating additional high-value indications. Along the way, as we've been preparing for phase 3, we continue to expand our staff to make sure that we are adequately resourced to conduct this very important study. So in summary, why Alterity and why now? Well, we are a phase 3 ready asset, and we're really aiming to deliver the first disease-modifying therapy for a terrible disease with no approved therapy. We showed up to 46% slowing of disease progression, something that's never been shown before. So really quite compelling phase 2 data on an endpoint. We've aligned with the FDA. We have a very robust commercial opportunity and a very differentiated profile with our drug, targeting iron to address the underlying pathology of not only MSA, but potentially of Parkinson's disease. So with that, I will conclude, and I'll be happy to entertain your questions.
Thank you, David. Great presentation. We've got quite a few questions which have come through. So the first one is related to that estimated $2.4 billion TAM you just mentioned and the securing of the new US patent extending the protection until at least 2045. Can you expand on how that changes the commercial picture for ATH434?
Yeah, so I think firstly, when we started this program, it's maybe a bit of history. We had generated a lot of data in Parkinson's disease. And these data were published coming up on about 10 years ago. So we've always been interested in Parkinson's disease, but it's taken us this long to get to phase 3 for MSA. And MSA is an orphan disease. And orphan disease, once the drug is approved, you have 7 years of marketing exclusivity, so a generic company can't market your drug. So based on that orphan exclusivity and given our current development timelines, we estimate that we'll have protection just based on orphan designation in the US out to 2037 or 2038. So the patent and that peak sales estimate of $2.5 billion or $2.4 billion is based on losing that orphan exclusivity in 2038, call it. So the patent actually gives us another 7 years on top of that orphan exclusivity. And you can imagine if you're at $2.4 billion at 2038, what with continued growth, there's going to be a substantial increase in that opportunity just in MSA. The other opportunity, obviously, that we've talked about a couple of times is Parkinson's disease. Because we didn't have sufficient patent protection at the time, we didn't pursue a Parkinson's indication. However, this new patent really makes Parkinson's disease a legitimate indication for us to pursue. So now we can really marry the commercial opportunity with the wealth of data that we've generated.
Right. And with the FDA aligned on that phase three design now, what's the realistic timeline from here to trial initiation, and what are the key milestones along the way?
Yeah, so we've been pretty clear that we expect to initiate trial activities by the end of the year, whether or not that's having drug at a site and having a site that's all IRB approved and ready to enroll, or screening a patient. We're not quite sure how far we'll get in the process, but we certainly expect that we'll be enrolling in first quarter of next year. That is the plan. Up till then, we'll be trying to get regulatory approval in other regions beyond the US. We will obviously make announcements regarding that. And then obviously the first patient to be enrolled is going to be a critical milestone because it sets the clock for the overall delivery of the program. And along the way, we have important scientific presentations coming up in the next several months. And we are really, working hard to publish the phase two trial data that I discussed. So the team is working very hard to get that study up and going.
Understood. And where and when will the phase three be held? Is this in Australia, and who will run it?
Yeah, so our goal is to, because this is a rare disease and we're aiming to recruit 200 patients, this needs to be a global study.
Right.
So we'll be recruiting centers in the United States, Australia, and then several European countries. And that final country selection, site selection, is ongoing right now. So that's one of the key things that the operations team is working on.
Mm, mm. And in regards to that phase two data, that was very strong on the primary endpoint. What gives you the confidence that that translates into a positive phase three result?
Yeah, well, that's a great question, Wei. I think development in MSA is difficult. Many have tried and many have failed along the way, both academic groups as well as industries of pharmaceutical sponsors. However, we used, I think, what is a unique approach towards patient selection. I talked a lot about that natural history study. That natural history study allowed us to select patients with a higher degree of precision than is routinely done in many of the trials that have not succeeded. So based on that, and the fact that we have chosen a dose that has demonstrated efficacy in phase two, we also plan to use a very similar, if you will, white glove approach towards selecting patients. We're going to vet every patient that comes into the trial. We're going to use these biomarkers that we used in phase two. And importantly, the phase three study is not that much bigger than the phase two study. The phase two study was about 75 patients, and phase three is just about 200. So it's about a 2.5 to threefold increase, which is not a big step up. Sometimes trials can increase from phase two to phase three by tenfold. And I think when you go to a large number of sites, you may lose control of the study. It's not as easy to have careful oversight. So we knew that, and that's why we're taking a very careful approach in just enrolling the minimum number of patients that we need to feel confident to show efficacy and to generate enough safety data that the regulatory authorities will have enough data to make judgments about the risk-benefit profile.
Yes, that makes sense. One of the slides that you showed before, slide 17, it talked about that phase two benchmark being a 48% slowing of disease progression. What would failing to replicate that percentage mean for the phase three trial result?
Well, so we have, we've designed the phase three study to have adequate power to detect that similar treatment effect of about, we saw anywhere from 2.7 to 3.7 points. So we have, when you design a study, you obviously use the prior results, the prior efficacy magnitude of the treatment effect, and the prior variability in the data. So we've selected a sample size that's going to give us 90% power to detect that treatment difference of that same magnitude. So I think the combination of a very similar patient population to what we recruited in phase two, as well as having an adequate number of patients, gives us the confidence that we can succeed again.
Okay. And when we look at the results for the phase 3 when they come out, what would be the minimum clinically meaningful percentage of slowing of disease progression needed to be considered statistically significant or a successful phase 3 trial?
Yeah, well, it's a little hard to predict what the actual percentage of slowing is going to be because it's based on how much the placebo deteriorates.
Right.
But if you looked at what we saw in phase two, the one dose group, the 75 milligram dose group, demonstrated about 35% slowing. So I think rather than look at what percentage of slowing is clinically relevant, I would look at the absolute magnitude. And I think a 1.5 point difference on the MSA rating scale is clinically meaningful. And that's really what would be the minimum expectation that would support a new drug application and an approval.
Mm. There's a couple of questions which have come through regarding funding. So can you please provide an update on any developments in relation to partnerships and funding of phase 3? And then another
Yes.
one,
Oh, go ahead.
and then the other one is just whether there have been any considerations to apply for a grant from the Medical Research Future Fund to support the phase 3 program.
On the funding opportunities, we are always interested in non-dilutive funding, as was just mentioned. While we haven't sought funding from the MRC, because I don't think they typically fund studies. These are relatively expensive studies to do, and I don't think the size of the grants typically will support that. But having said that, we have had other academic groups that approached us to study our drug in other diseases, other orphan diseases, as well as even in Parkinson's disease, where they would fund most of the trial. So there are real opportunities for non-dilutive funding that we are evaluating. Having said that, from a partnering standpoint, we've had very active partner discussions since we had our end of phase 2 meeting, and we are pursuing those discussions actively. You never know exactly how those discussions are going to go. It's a slow time right here in the Northern Hemisphere in the US, where many of the partners that we're talking to are based. So we expect in the near term to have clarity on that. But there has been significant interest considering the substantial commercial opportunity, as well as given the robust data. And I would say that the new IP, the new patent surrounding the composition of matter patent is quite important for some of the prospective partners that we're talking to because Parkinson's disease is one of their main interests.
Yeah, definitely that extension makes it a lot more attractive. Coming back to Parkinson's, so you flagged this as a disease, the Parkinson's disease as a future opportunity. So how should investors think about that without it distracting from the near-term MSA focus?
Well, Parkinson's disease is a completely different program and would take significant resources to conduct. So we're not going to do anything that's going to get in the way of starting the MSA phase 3 program. That is far and away the most important value driver. But that's where the prospective partnership discussions are so interesting because such a partner could potentially have not only the financial resources, but also additional human resources to dedicate to the program. Our know-how, we obviously understand the drug. We understand how to evaluate it in the clinic. So there would be important synergies there. But I don't see clear activity on a Parkinson's disease program occurring until we're well into phase 3 next year.
Okay, makes sense. Final question. What catalyst should investors be watching for between now and the end of the year? And what would you consider the most important one?
Well, I think when we are ready to enroll, when we announce that we've initiated clinical trial activities, I think that is probably the most important milestone. As we've talked about, there are several other things going on right now, but commencing this trial is far and away the most important milestone ahead of us, because that will mean that we have selected, all of our vendors, that we have regulatory approval, we have IRB approval, the drug is at the site, and there's a lot of things that have to go into having the trial ready to kick off. So I view that as the most important thing. But then not far behind it, obviously sharing more of the phase 2 data, both in public form, full publication form, as well as presenting it at scientific congress is really important as we help people understand the potential of this drug to treat these devastating conditions.
Hmm. Hmm. Well, it sounds like you've got your work cut out ahead of you, David, but a very exciting time. And, with that extension, we see a lot of excitement in the company. So thank you, Dr. David Stamler, for the time today. It's been a pleasure.
Thank you.